Awesome. Hey, everyone. I'm here with Dr. Jensen, who prefers to be called Lars, which is pretty awesome. He's joining us from Denmark, and he is just back from a flight that was delayed till 1 a.m. So really appreciate you hopping on a call with us. I'm Rahul, founder of Julius, and Lars made a video about Julius and using Julius for some bioinformatics work. And I reached out to him. I said, thanks so much for the video, and thanks so much for finding areas where Julius could get better. How about we, kind of make a video and show folks how this can be used with the string database. Now, if you're in bioinformatics, you've probably heard of the string database. And Lars is one of the creators of that whole project. So I'll let Lars introduce himself.
Yeah, I'm Lars Juhl Jensen. I'm a professor at University of Copenhagen, where I run a research group in bioinformatics. And one of the main things we're doing is tool development. And the most famous tool of those is the string database that we developed together with groups in other countries, especially the group of Per Borg in Germany and the group of Christian von Mering in Zurich in Switzerland. So very quickly, the string database is a database of protein-protein interactions and associations. So you can go in and look up genes or proteins of interest and get a network that tells you which ones work together. And that's something lots of experimentalists use for analyzing their big data sets and also for visualizing the data sets onto networks. One of the challenges you have is, of course, that as a bioinformatician, you are very often asked to help with things that, from a research standpoint, as a bioinformatician, is not particularly interesting because it's the kind of standard analysis, but since most of your collaborators working in a wet lab have never written a line of code, they need your help to do it. And I think that's where Julius AI is interesting in a future perspective, that a lot of those trivial things, I hope they will be able to just do themselves with a tool like this and not have to bother us.
That's awesome. So I guess what Lars is saying is there's a lot of boring work. Sometimes it's mundane and day-to-day, and you can probably automate that, hopefully with Julius AI. But Lars has had a time to play with Julius, so I'm just going to let him take it. Feel free to share your screen, Lars.
Yep, just did.
Awesome.
Just need to move a few windows here. Okay, I think we're good to go. So what I have here, you're seeing the window now?
Yep.
I hope. Yeah. Is a big data set. It's one that we were working with experimentalists right next door in Copenhagen. And it's a so-called phosphoproteomics data set. So what that means is that you've used mass spectrometry, which is effectively a very fancy, very accurate way to measure which proteins get modified through so-called phosphorylation under some conditions. In this case, looking at certain types of cancer. And when we've loaded this spreadsheet, you will see that it contains a lot of different rows, I think 700 and something rows, and each of them represents one modification site in a protein. So in this case, you have the proteins and corresponding genes. Proteins are identified with UniProt identifiers. And then you have a modified sequence. You have the position that is modified. And then you have measurements under 3 different conditions. You have so-called log fold change values comparing different conditions with each other. And we have some p-value telling us whether this site in this protein is significantly regulated. And although here the first 5 sites are all on different proteins, since proteins are long entities, you can of course have multiple different places within the same protein that is modified. So we could start by just asking something very trivial, like saying, how many sites are significantly modified at a 1% level? So being statistically significant at a 1% level. So that's just a matter of filtering on the column called p-adjusted, and then based on that, afterwards counting how many sites are left. So that should be a very easy thing to do. Now, the slightly more tricky thing is we could, of course, then just ask how many unique proteins do these correspond to? In which case you would just need to take the rows that survive that p-value filter and then go and look up either the column called UniProt or the column called gene name and do a unique count. And as you see, it's 238 proteins that have at least one significant site, and there are 343 significant sites, so some proteins have multiple significant sites. So these are sort of very simple things that obviously I would guess, I mean, most of the experimentalists are pretty good at Excel, so they could probably do this in Excel themselves. I would argue this is the easier way of doing it, but they could get this done. What you often do in when you analyze proteomics data is you make something called a volcano plot, which is a plot where you plot the log fold change values that I talked about before on an x-axis, which is how much does a site in this case change. And on the y-axis, you plot the minus log 10 of the p-value. So you're simultaneously showing how significant is the change and how big is the change. So let's say make a volcano plot based on, let's say based on the column, based on the log fold change values from EOC versus FTE. You will note here, I didn't tell it at all what a volcano plot is or what it should put on which axis. I only told it the thing that it will need for the x-axis. I didn't say anything about what to do with this.
Cool.
And
go ahead.
No, feel free.
I was just going to say, hopefully this works because I just pushed some code last night. And so there's some other engineers. So maybe it breaks, maybe it doesn't. We'll see. This is completely unscripted. We're playing live with it.
Yep. I have seen this problem before, so it did indeed make a mistake here. So what it did, so it did put the right things in principle on the axis, but it didn't understand that what we have in the column EOC versus FTE is not ratios, but log ratios. So it log transformed the data that were already log transformed. So I guess if I tell it the column contains log2 transformed data, not raw ratios. And it looks like it figured it out that indeed it should not do a log transformation since it now knows that it's already log transformed. And we should get a more symmetric looking plot. And that looks right. So then what you would typically want to do is to put some cutoffs on it and say something like highlight the sites that are significant at a 1% level and change at least twofold. So that should be the ones that have a log p-value of at least minus log 10 of p-value being at least 2, because that would be a 1% level. And at the same time, actually something is a bit funny here. I made a mistake here that I didn't catch. So it made the plot, I'm pretty sure, of these 343 or however many significant sites we had.
Yeah.
It Which I can understand because this is a conversation, right? And I asked it to figure out which ones are significant first, and then I asked for a plot, and then it made a plot of the ones. So please include all sites from the Excel sheet, not only the significant ones. Doesn't help that I write call when I mean all. Can I stop it? I guess I can.
Yes, and you can also edit the message.
Okay, let's try that. I mean, I wouldn't expect the right thing to happen if I write the if I tell it the wrong thing. Okay. Oof, now it got confused.
Yeah, I think we could stop it. I think it kind of overlaid the two messages.
Yeah.
So if you hit stop one more time. Oh, is that what you want?
Let's see. No. It got rather confused here.
Okay. We can
it seems to not be very happy stopping.
Yeah, if you could just hit stop. Yep, there you go. And sometimes it doesn't stop. And this is beta product. So we'll fix. I'll go back to this bug with the team. But if you at some point in time in the message where you ask it for the plot, you can just say edit that message and say, can you give me
yeah, so maybe if I go up in this one.
No, if you just scroll up.
No.
Like whatever your message is.
There, yeah. So what, should I edit that one?
I guess you could say
then also include all sites, not only the significant ones. And then hopefully we will be able to continue with highlighting the ones that are significant and change at least twofold. All right.
Awesome.
And then I think I will just did I now lose the other one? Yeah, okay, fine. I can just say
how long has the string database project been in the works?
Forever. I got involved in it when I started working at EMBL in Heidelberg in 2003.
Wow.
And back then they were probably already on version 5 or something like that.
And what version is it on now?
We just released version 12 recently.
Cool.
Okay, let's see. Are we in luck here? Yeah, this looks right. So now you see all the red sites are above the y equals 2, which is a 1% p-value cutoff, and they also all change at least twofold, meaning that they are outside the either +1 or -1. So twofold up or twofold down. And something that has definitely worked nicely in the past is add dotted lines for the cutoffs. you'll typically want to show where your cutoffs are. And I guess the next thing we would want to do is then get a list of the proteins that correspond to these sites, because now we're still looking at phosphorylation sites, and we've now found the ones that change enough and are significant, and then say, give us a unique list of all the proteins for those, and then let's see if we can get a string network. Would that be the battle plan?
That sounds fun.
Good, we got the cutoffs in here. Then, how many sites were highlighted in red?
Also, one thing I want to show real quick, and I know you mentioned code in the past, but if you click finished working, it should show you the Python code it's writing now.
Yeah, and now it also says show code. Yes.
And it's easy for, if someone knows Python, it's easy for them to verify the results. And
exactly.
Yep.
Good. So that gave us this, and then we can say how many unique proteins do these correspond to? That should then be 200 and something. Then I should be able to ask for just give me a list of the UniProt identifiers for these. Yeah.
Awesome. Cool.
So now we have our proteins, and I guess we should be able to get a string network. So if I hop over to the string database, then maybe just to give you, can I do this? Yeah, probably. So the quick and dirty way for biologists, if they don't need to analyze the network, but they just wanted to actually get a network of these proteins.
Yeah.
Then, oh boy, is there a way to make it sort of speed up a bit instead of seeming like it's typing it all?
That's a great question. I wish there was. And what we can do in this case is bypass the AI and have it write a script. But I think what it's doing is AI is manually going through each of them and trying to write them by hand. This can be frustrating.
Because obviously it would have been very easy Python code to just dump all the elements of this list, right?
Exactly. This is good feedback. I mean, in a way, this is like a user interview too. And Lars, I really appreciate you, poking with Julius, finding areas where it's good at, finding areas where it needs improvement. And I guess we could maybe use a second to talk about the data ethics point you had made in one of your videos. If you could share a little bit more about that, would be awesome.
Yeah, so I was just playing around after I got a license from you. Thanks for that, to play around with it freely. I started playing around also seeing, would it be happy to help me do things that you definitely shouldn't do as a scientist? Because you probably run into sort of some of the guardrails that you have on ChatGPT if you've ever used ChatGPT. So you ask for something and it says, no, I'm not going to give you that because that's not okay. And in this case, I asked it to basically make up data. So I pretended that I had some data set. It was anyway fake data too, but I had some small data set. I only had 2 biological replicates, which is clearly not enough. And then I asked it to make up a third. And then it looked at the distributions of the numbers from the two that existed and then generated random data that followed the same distribution. Then I asked it to do statistics on that, and it happily did statistics on all the data, including the data it had just made up on its own. And yeah, it was way too easy. Then for one of the genes, it was still not significant. Then I asked it to make it significant, at which point it just went in and started fudging the numbers to get a good p-value. This is the kind of stuff that I'm sure there are people out there who've been doing with pen and paper for decades, cheating their data to get a significant result for something that wasn't significant. But it's definitely not okay. And it would be nice if the tool at least sort of came up with a warning being, what do you think you're doing?
Yep, totally. And I appreciate you stress testing it. This is really good feedback for us to go and work on. And
so now we have the big list of UniProt. So if you just jump over quickly to string, just to show you what it is we would like to be working with, here's a long list of proteins. One per line or CSV. Oh, joy, I should have had commas between them. I don't think we want to wait for that. Let's stress test the web interface and see how it behaves. Why not? If I put them like this, is it? Oh yeah, even though it said it had to be comma separated, it happily took it space separated anyway. We are in luck. And here you got in the string web interface, then a protein interaction network of all of these. You have many different colors of lines representing basically different sources of where we got the interactions from. But I could now very easily just go in, change it a little bit. Let's make it maybe a physical network. Confidence view, getting a network like this. And I guess this is the kind of thing we might want to try to get into Julius as an actual network to be able to work with it. And I guess that should be possible because String has an API. So maybe starting with just getting the image. So should I just try to copy the example call?
Yeah, totally. And would you want to do this in the same chat or a different chat? Is there something we want from this chat that?
I think we're done with this, except from having the protein list.
Perfect. We could, I guess, make a new chat and say, hey, I want you to.
How do I transfer this whole list to the new one?
Oh, you want to transfer? Then let's just stick with this chat if you're going to use.
Yeah, because I need to work with this list.
I see. Let's keep it in this chat then.
So hey, how do we go? Do we just give it an example? Here's an example API call for fetching a network image from the string database.
Yep. And I guess we would want to tell it what we want to do with it.
Yeah.
I think it's just, yeah, we could hit stop there. And
yeah. All right.
Edit our previous message and say what we want to do with the API. Do we want to get a network?
I was hoping I could just do that in a separate one, but all righty, we can put it here. Use the 233 proteins to retrieve a network. They're optional. We shouldn't need them. So I hope it will maybe figure out to put the identifiers in there.
And if it doesn't work, I guess we could, from the API docs, just copy a paragraph. And we could just dump it in there and then see if that works. go.
I think we got an error because at least my image viewer says that the PNG file isn't a PNG file.
I see. Okay. We could go back to Julius and maybe clarify the instructions a little bit.
Yep. So we just copy.
Yeah, I think that should be good. And then dump it in there.
Here we go. Close this one here.
Yep.
Do we take this one and edit it? It also it didn't really have a chance here because there's no way it could have known that you're supposed to separate the different proteins with this, which is the URL encoding of a new line.
Yeah. Yeah.
Let's see if we have more luck. 2.3 megabyte instead. That sounds a whole lot better. Yes. It's of course in the wrong window now. So if I just... Or could you see it?
I could not. I just see your browser window.
Yeah, exactly. Because I'm sharing the browser window. That's what I was thinking. But if we just over here and throw this into the browser window, there you have it.
Wow.
So that's the file I got. And as you see, it looks very, very similar to what we had in the web browser when I copy pasted the same identifiers in. In fact, it should be the same.
Awesome.
I guess, should we see if we can make it a... What does it have up here? Chuck. It has something about the network type, physical. Let's see if we can figure that out.
Let's do it.
Instead, and increase the confidence cutoff to 700.
Awesome. I'm excited. While we do that, I would love to take a look at the code for the last step. I'm assuming it.
So this one.
Correct.
Set species to human. Sure. Yeah, so it joined all the uniprots with %0d. And then put that on the base URL so that.
And I also like that there's
right. And then let's see what it did here. Params equal params. Set network type to physical, set required score to 700. That looks right. I would expect to get the right network. Let's see it. If my browser will collaborate. Yep. Yeah, that looks right.
This is great. I guess one more thing I want to clarify, I guess could be useful for non-technical users is the code has comments in it, and the comments could be useful for users who are non-technical to just quickly verify the logical steps that the AI is going through, and maybe perhaps identify things it could fix if the output is not expected.
Yep. Let's see if we can. let's see if it figures out on its own that it simply has to, in the base URL, replace image with TSV. Let's see what it did. Yep, it changed the base URL to be API TSV network. Can I? Load this network as a network X object. This, of course, requires knowing a little bit about Python, that's the thing you would want to use. But
yeah,
I guess if it has a TSV file, it should be able to load it.
Let's give it a try.
Yeah. I think what would be really cool would be to be doing a little bit of some sort of prompt engineering to be able to have some standard things one could paste in and be like, I want to do string networks in Julius, and then have it automatically have been taught the whole API, and now it knows that, and you could do simpler prompts. I'm sure that should be possible, because since it's a conversation, one should be able to have that as sort of a preamble.
Right. Absolutely. And I would love to show you a feature for that. We kind of have in the works. Haven't stress tested it, but should somewhat help with the thing you're describing. Awesome.
Absolutely. If we get... Okay, it didn't quite get the point that I wanted a figure. Okay, visualize this. Oh, maybe it was just still busy and didn't get it. Let's see.
Oh, one more thing I want to clarify is if you see the gear icon on the top, right?
Yeah.
That icon.
Okay, so okay, that might be the world's worst network layout, but yeah, all right.
We could ask it to space it out. You can say, hey, the text is too big and it's overlapping. I cannot read it. Could you space it out?
Yeah, but it wouldn't manage with 233 anyway. It's sort of an issue here that also now you have the whole thing in string identifiers, which are really long instead of gene names, which is what you would want to show. So really what we should have done is, and that's also why you'd want to have this preamble, is to really first use the resolve functionality to map the identifiers so that in the data frame with the proteomics data, you would get another column that would be the string identifier, and then use that for fetching the network so that you would still have everything linked and you could put sensible names on it.
Yeah.
Also, because the next thing you would probably want to do was to color code the proteomics data onto the network. And right now, things are completely disjoint because in the data structures that I now have in the notebook here, I have a network where things live in string identifiers. I have a different data frame that has things in uniprot identifiers, and I don't have a mapping between them because that mapping was done over at string and I never asked to get it.
Yeah.
But that would have been a longer process to do, so that would have been too long for this online session.
Yeah.
I'm sure it's doable.
Totally. I guess we're out of time now.
Should I click the gear symbol up here? There was something you wanted to show me.
Yes, exactly. And this is where you could set your specific instructions, where you say, okay, biology, you could give it more specific things like, I'm a bioinformatics person. I need you to explain things to me in an analytical way. And this is a feature in beta. And
this stuff is going before, so basically whenever you open a new chat.
Exactly. And you can basically set the preamble where like, hey, this is the context of this chat. And then keep moving.
That would be sweet if, so I mean, you have these ones, but I assume where you're going next is to allow people to make custom ones themselves.
Exactly. And you can also,
instead of just being able to choose your pre-made ones and edit those, it would be nice to be able to, make one called network biology, right?
yeah, totally.
And then we could, as a user, one could go put in the stuff that is needed.
Exactly. Great. Cool. This is awesome. Thanks so much for hopping on the call, Lars. I know this is super late for you, and
it's not too bad. It's not 8 o'clock in the evening yet.
Well, you have been up for a long time. And
yes.
But as you all can see, Lars has a way better camera and mic than I do, and a really cool background, which means he must have a YouTube channel, and he does. So I'll have a link in the description for his YouTube channel, which is where I found
Thanks a lot.
a bunch of really cool videos. I discovered the Julius video there, but then turns out Julius is just the latest video he's made. There's a bunch before that. And I saw a bunch of those and learned a lot about network biology. I'm not a network biologist, by the way. I'm a computer science person. So definitely check it out. He has a Twitter that's pretty active, and Instagram, and have the links for all of those in the bio. And Lars, really appreciate you taking the time to just walk us through how you can play with bioinformatics in Julius.
Well, thanks a lot, Rahul. It's always great fun. I've enjoyed our meetings, also the earlier ones that we were not sharing with people.
Yeah, totally.
Have a nice day.
You too. Bye-bye.
Bye.